Welcome back, folks. This is Chapter 8, Part 1. In my opinion, one of the easiest in the whole book to understand. We begin a journey away from DNA into RNA and proteins. The very first step is the conversion of information from DNA into RNA. And that process has a name. It's called transcription. With this video, we'll look at some general properties of transcription, but focus on bacterial transcription, and then in the next video we'll look at eukaryotic transcription. Since the discovery of the structure of DNA, the attention of scientists turned to RNA. They wanted to know how proteins were made from DNA. In particular, they knew that DNA was located in the nucleus of eukaryotic cells, and they also knew that proteins were synthesized in the cytoplasm. Neither of these two components could exchange places, so there was some other mechanism available to the cell for passing knowledge from the nucleus to the cytoplasm so proteins could be made. Before we understand the parts to transcription, let's have a quick look at the structure of RNA, which is common to all living things. RNA consists of building blocks, just like DNA. However, in this case, they're known as ribonucleotides, not deoxyribonucleotides, as was the case with DNA. Luckily for us, we just have to remember the two differences that exist chemically between DNA and RNA. So whereas DNA contains A, C, G, and T, RNA contains uracil in place of thymine. So for DNA, we have ACGT. For RNA, we have ACGU. Scientists are not really sure why this difference exists. It could be a legacy of the evolution of DNA as a slightly more stable form of information molecule. The second chemical change is the presence of an atom of oxygen, which is not present within the sugars of DNA. And those changes are both indicated on this panel. You can see this is uracil, and in uracil we have certain differences from thymine. If you compare this to DNA structure, you'll find that in thymine there's a Ch8 here rather than just a hydrogen. And that makes this base very similar to thymine in that it can base pair with adenine. The second change is right here on every single one of the sugars that are used for constructing RNA. This oxygen is present in RNA and will be absent in DNA. And if you recall your chemistry, this will be the second carbon on the ribose. These changes may seem minor in that the backbones of the two types of nucleic acid are nearly identical. The only difference, of course, is the presence of the oxygen in RNA and its absence in DNA. The other similarities are that the subunits are placed together by the enzymes, the polymerases, using phosphodiester bonds between adjacent nucleotides. So that's a place where you should revise and figure out what a phosphodiester bond is, please. And the last thing is that complementary base pairs do allow different nucleic acids to line up. The only change is that A and U pair up when DNA and RNA are concerned. Students are highly encouraged to use this type of figure to revise everything they've learned so far. One thing is to look at the orientation of the DNA molecule on top, and look at the orientation of the RNA molecule being synthesized at the bottom. As we told you in the past, nucleic acid synthesis always takes place backwards. So from 5, 4 to 3. And this is confirmed here by this figure. DNA is always read forwards, 3, 4, 5. So the top strand, even though it goes that way in terms of orientation, it is read in this direction. And of course, a phosphodiester bond is right here. You can see the phosphate group of the incoming nucleotide, this one here, is used to bond itself to this hydroxide of the sugar of the previous nucleotide. So the phosphodiester bond refers to the presence of this oxygen. Right? A couple of other major differences between RNA and DNA is that RNA is normally found in single-stranded form, whereas DNA is normally found double-stranded. Being single-stranded doesn't mean that it occupies a one-dimensional place in space. The RNA molecule has a tendency to form localized secondary structure. That means parts of the molecule that can form complementary base pairs with itself will snap back on itself to form various structures. So in that sense, RNA resembles proteins more than it does DNA. In fact, RNA may have been in the past, we suspect, a dual function molecule in that it carried information just like DNA does today, and it also had secondary structure just like proteins do currently. The other major difference is that the enzymes that life uses to copy DNA into RNA are different to the ones that life uses for copying DNA to DNA. The only difference is in the name. These are called RNA polymerases, not DNA polymerases. But they do, as we mentioned earlier, exactly the same thing. They take ribonucleotides and they add phosphodiester bonds, but only to the 3' end. Now, here's another major difference. Whereas DNA polymerases must have an existing 3' end in order to start building further nucleotides, RNA polymerases can start without having a 3' end. So they need fewer rules in order to initiate RNA synthesis. If you recall in DNA replication, that's exactly how the RNA primer is laid down inside a replication bubble. Returning back to the historical perspective, we need to make sure that students are familiar with the pulse chase technique, which was mentioned in previous chapters. So here, the experimenters use radioactive materials, or these days fluorescent materials, to generate a chemical pulse within the system that they're studying, cells or individual machines of transcription, as in this case, followed by either the removal or the dilution of the radioactive material with normal material, non-radioactive material, or non-fluorescent material. And then they can follow through time-lapse processes where the radioactive material ends up moving as a part of an escalator pathway. Using pulse chase experiments in bacteria in 1957, scientists were able to trace the relationship between bacteria and their infection with bacteriophages. These experiments showed that immediately upon infection of bacteria by these viruses, there was a burst of transcriptional activity, which quickly dissipated as the RNA molecules were broken down in the cytosol of the bacteria. Two major conclusions were recorded. One was that RNA transcription was necessary every time before new phage proteins could be formed inside these bacterial cells. Additionally, the RNA was associated with ribosomes during this process. Similar experiments in eukaryotes yielded a slightly different outcome. First, the radioactive material, in this case uracil, was located in the nucleus of all cells. After a short period of time, the radioactivity was seen to migrate from the nucleus to the cytoplasm, before it also dissipated, but this time period was much longer than in bacteria. The conclusions, which are not surprising to us today, were that RNA synthesis began in the nucleus and then migrated into the cytosol, where the ribosomes are located. Both these sets of experiments relied on some kind of molecule conveying information from DNA, where it's stored, to the ribosomes, where proteins are made. So naturally, scientists labeled this molecule messenger RNA. Let's turn our attention to the different classes of RNA that have been found inside cells. Your textbook explores a few of these in this chapter. However, if you look online, for instance at Wikipedia, you will find there are over 40 different types of RNA discovered at this point in time, with many more hoping to be found shortly. Let's understand 6 of those at this time. The first class is messenger RNA. Messenger RNA is produced by cells in order to subsequently produce proteins from the information in the genes on the DNA. Messenger RNA, by comparison, is relatively short-lived inside cells. There is only one type of RNA that undergoes translation, and that is messenger RNA. It doesn't matter whether it's eukaryotes, prokaryotes, or anything else. It's only one type of RNA that interacts with ribosomes and produces protein. All the other types do not do this. And transcription of RNA is often followed by additional modifications to those RNA molecules, especially in eukaryotes. And that process is called post, that means after, transcriptional processing. All the other classes of RNA are called functional RNAs because they perform some function within the cell. Functional RNAs do not encode proteins. Only messenger RNA does that, so we must understand that. So common to eukaryotes and prokaryotes are ribosomal RNAs. These functional RNAs combine with many proteins to form the structure of the protein-making machines called ribosomes. And all cells have ribosomes. Transfer RNA, which is a subject of another chapter, but we still talk about it here because it's very common. Transfer RNA is also a functional RNA. It has a function of carrying amino acids from different parts of the cytosol to the ribosomes where it's needed for making polypeptides. Some of the other classes of RNA may be found in particular branches of the tree of life. So these are only found in eukaryotes, as far as we know. So linear chromosomes, such as in humans, have to be replicated using special enzymes near the end, and those enzymes carry within them RNA. And that RNA is called telomerase RNA. So this is part of the telomerase complex. And we know from a previous chapter how that works. There are also discoveries of small nuclear RNAs, snRNAs, of various types. The category is growing as research is being performed. These small nuclear RNAs are again only found in the nucleus of eukaryotes and not in prokaryotes, and they have a role in messenger RNA processing, this post-transcriptional processing that we spoke about. Additionally, in plants and animals, but not other types of eukaryotes, we find micro and small interfering RNAs, so MI and SI RNAs. And these are involved in more regulatory types of processes where they control the activity of messenger RNA in a pretty complex way that we'll get to in the future. The summary of the types of RNA just discussed is given here, but students should try to understand which are present in which branches of the tree of life. So most of these are found only in eukaryotes and not prokaryotes, and that's important to understand. Let's now focus for the rest of this video on transcription in prokaryotes, namely E. coli. That's where most of the research was conducted. So one thing to understand is that transcription itself is a 4-part or 4-stage process. It involves recognizing where to begin to copy a gene, how to start copying the gene, how to continue to copy the gene, and when to stop copying that gene. This logically makes sense. You don't want to copy all the genes all the time. That would be a waste of energy and resources, and not necessary. So you only copy the genes that that cell needs at that particular time. If you recall from previous courses, cells basically are chemical sacs which respond to their environments. So if the environment requires you to produce a certain set of genes, those genes will be activated, other genes will be kept silent. Just to reiterate the use of certain terminology used in genetics and biochemistry. So the term template strand refers to that strand of DNA which is physically interrogated by the RNA polymerase and copied into a new RNA molecule. It's also called the non-coding strand in certain textbooks. The strand that's not used by the RNA polymerase but pushed to one side is called the non-template strand. However, in the same textbooks as them using the non-coding strand, they sometimes refer to that as the coding strand. And in fact, in genetics, the most common label is the coding strand, as we'll see for obvious reasons later on. One of the requirements of any student of genetics is that they are familiar with how genes are referenced by other people. So whenever you have a single strand of nucleic acid, we always label it backwards. So 5, 4, 3. And that makes sense because most things are made in the same direction. Here we have the 5' end, also known as the beginning or the upstream end, and we have the 3' end known as the end, and that's also known as the downstream end. We'll see that on the next slide. The important thing is that the messenger RNA is made from a particular location on the gene. It's always made from the +1 nucleotide, even though nucleotides ahead of that, which are numbered minus in an increasing fashion, do exist within a gene. The messenger RNA always begins at +1 by convention. Since we are focusing on bacterial genes in this section, we can make another generalization that for most bacterial genes, the switch that controls the activity of that gene is located directly in front, upstream of the +1 site, and that is called the promoter. The analogy to a light switch may be useful as we go through subsequent chapters. As there may be in your house two types of switches, a direct on-off switch and a dimmer switch, we find similar examples in biological situations. A few genes have an on-off switch. Either they're on or they're off. When they're on, they're 100% on. When they're off, they're 0% on. But most genes are like a dimmer switch, where the promoter may be on by a certain amount. And if it's on by 50%, then that gene is active at 50% its maximum rate, or it could be on at 70%, depending on the needs of that particular cell at that particular point in time. In addition to the promoter, the next part of a gene is called the coding region of the gene. Nothing to do with the coding strand. This is a different use of the word coding. So the coding region of the gene makes the messenger RNA, from which maybe a protein may be produced in the future. The last region of a gene is called the termination region. This is where the DNA carries information to tell the RNA polymerase not to go any further and detach itself from the DNA. Terminator sequences in prokaryotes are generally found downstream after the coding segment of that particular gene. This figure reiterates this information. So a gene consists of three parts so far: the promoter, the coding region, and the terminator. The promoter is the switch, the terminator is the signal to stop further progression of the RNA polymerase, and the coding region carries the information that's going to be converted into messenger RNA. Additionally, for this particular gene, the terms upstream and downstream are indicated, and these are analogous to a flow of water down a river, upstream being where the water originates, and downstream is where the water is heading. In bacteria, all bacteria, they have evolved a single type of RNA polymerase enzyme for copying all their DNA into RNA. There are no exceptions found so far. How do we know this? Well, we can use chemicals like antibiotics. Rifampicin binds to RNA polymerase and inhibits the phosphodiester bond formation capacity, therefore preventing polymerization. The binding of rifampicin to RNA polymerase prevents the production of all types of RNA, therefore indicating the presence of just one class of RNA polymerase. It's important to know the composition of the RNA polymerase in order to then deduce its function. RNA polymerase can be found in two configurations. One is alone as a complex of polypeptides, known as the core enzyme. The core enzyme consists of 5 polypeptide subunits, 2 alphas, 2 betas, and an omega, and that's called the core enzyme. There exists another subunit called the sigma subunit, which can combine with the core to form the complete enzyme, also known as the holoenzyme. And you'll see the difference in a few seconds as to the importance of the two parts. Experimentation has proved that the core enzyme can bind to promoters, but with very, very low efficiency, in most cases activating the gene at just a few percent of the maximum rate. Figure 8.4 shows the two parts. Here is the core enzyme, and here's the sigma factor. When the two come together, they associate in a complex fashion to form the holoenzyme, the enzyme that's very efficient at activating transcription. Why would evolution disconnect the RNA polymerase into two parts? The reason is that the sigma factor can be substituted out for one of several different types. So inside a E. coli cell, we find a handful of different sigma factors, and each sigma factor changes the shape of the core enzyme in such a way that it binds to different types of promoters. So different genes can be activated depending on which sigma factor is present inside that cell. All known promoters must be double-stranded DNA in order to allow the RNA polymerase to attach. An important concept in genetics is that a gene is a tiny fraction of the DNA upon which it resides. So a gene in an E. coli will represent a tiny fraction of its chromosome. So how does the RNA polymerase know where the promoter is and which way to attach to that particular gene? And the answer is that the DNA itself carries small, significant regions called consensus sequences, either at the promoter or close by, that the RNA polymerase recognizes when that particular DNA is active. Here is another rule in genetics to remember. Sometimes some texts only list one strand of DNA and not both. In that case, the rule is that you're looking at the coding strand, the one that's not used by the RNA polymerase, and the orientation of that strand on a piece of paper is from 5' to 3'. Comparison of many genes in prokaryotes reveals a pattern, and that pattern has a consistency that's not absolute, but within a small range, and we call that a consensus sequence, a consensus. So at the -10 position on a gene, there's a region that appears to be present in many genes, called the Pribnow box, after the scientist who discovered it. But sometimes you just find it as the -10 sequence. And the most common iteration of that sequence is T-A-T, A-A-T. T-A-T, A-A-T. You may want to remember this. And also, just a few nucleotides away at -35 is another consensus sequence called the -35 sequence, which has the 6 bases of TTG, ACA. TTG, ACA. Why is this important? Well, the RNA polymerase is a pretty hefty molecule in relationship to the DNA that it interacts with. The RNA polymerase has, for instance, two hands, just like you do. And each hand grabs one of these two locations on the DNA. The first hand grabs the -10, and the second hand grabs the -35. And the enzyme occupies this entire region here, as your body would if you were to sit on this. This figure tries to relate what we learned previously to what we're learning now with the sequence of the promoter. The second of the four-stage process of transcription is initiation. Initiation itself is a two-stage process where the RNA polymerase interacts with the promoter in two subsequent events. The first of these is a loose attachment to the DNA called the closed promoter complex. That's indicated here in this figure 8.6. The second step is the enzyme able to unwind about 20 base pairs of DNA at the -10 consensus sequence of the promoter, forming an open promoter complex, indicated here in figure 8.6. Once the hydrogen bonds holding the DNA duplex together, the duplex simply means the two strands of DNA in the DNA helix. Once the enzyme complex melts those, the strand that's going to be acting as the template is picked up by the enzyme, and the next phase of transcription begins. We alluded to the fact that E. coli has a number of different sigma units. The most common one is called sigma 70, according to its molecular mass. There are so far recognized three others, which are sigma 28, sigma 32, and sigma 54. And scientists have determined through experimentation when and by how much the other sigmas may be active in transcription. When E. coli cells are subject to alternative environmental cues, i.e. conditions, they will then start manufacturing alternative amounts of these sigma factors. For instance, if an E. coli cell experiences additional heat in its environment, it will then start making sigma factor 32, because those will activate the heat shock genes, which then biochemically protect the bacteria from the additional heat. Other bacterial strains and species contain their own sigma factors in addition to these. The next phase is elongation. Elongation is quite simple. The enzyme continues its journey along the DNA. However, within a short distance of starting elongation, the sigma factor is no longer needed. It's done its job of opening up the DNA at the promoter, and the sigma factor dissociates. That means it falls off, and the journey is completed by the core enzyme itself. Once the RNA polymerase core enzyme reaches the end of the gene into the terminator sequence, the final phase, the final stage of transcription takes place, which is termination. Studying E. coli reveals two different mechanisms of termination. The first is called intrinsic termination. It doesn't require any external help from proteins or anything else. It just happens between the DNA and the RNA polymerase core enzyme. But for other genes, there's additional help necessary for termination to take place. In this case, special proteins are necessary to interact at this location and to stop the transcription of the gene further beyond the termination point. Now that's called Rho-Dependent Termination because the protein has been labeled as Rho, and it's very important. Intrinsic termination depends on sequences carried by the DNA, which once converted into RNA, allow the RNA to quickly adopt a hairpin structure just behind the core enzyme. That hairpin structure has a profound effect on the passage of the RNA polymerase, slowing it down, and then eventually pulling it off the template strand of DNA. Figure 8.7 sheds further light onto that process. So here's the terminator sequence that we are looking at from this point up to this point. And we can see within the terminator sequence there's a consensus. And the consensus is that there's an inverted repeat. Once that's coded into RNA, the RNA also contains the inverted repeat. Once the RNA is long enough, it will form this hairpin structure. And the hairpin structure will then have a mechanical effect on pulling immediately on the RNA polymerase, which will be in this region here. And as the RNA polymerase copies the DNA into this poly-U region, the poly-U region causes further slowing down of the passage of the RNA polymerase, and in conjunction with the hairpin is enough to prevent further transcription. The alternative method of termination, the Rho-Dependent Termination, only applies to certain genes, and as we said earlier, requires the presence of a protein called Rho. There's a lot of overlap between the Rho-dependent and the intrinsic system. As in the intrinsic system, the RNA polymerase will have to go through the terminator sequence, and it will form the hairpin at which it slows down, and also it will make a string of poly-U's in the RNA. The additional thing here is that just upstream of the termination sequence is a Rho utilization site, a special site on the DNA that then encodes the RNA. The RNA is then compatible with the protein Rho. Rho will bind to the RUT site on the RNA and then start traveling fast towards the downstream direction towards the RNA polymerase. And because the RNA polymerase has been slowed down by the termination sequence, the Rho protein will eventually catch up and physically dislodge the RNA polymerase from the DNA. Please take your time in understanding this. This is an important concept. One requirement is for you to know the difference between prokaryotic and eukaryotic transcription. As we mentioned, bacteria have a single RNA polymerase with multiple different sigma units that allow different promoters to be attached to and activated. Eukaryotic cells have evolved 3 different RNA polymerases that transcribe different sets of genes, and we'll see that in the next video. In addition, the eukaryotic RNA polymerase that is used for most protein coding is different in many ways from bacterial RNA polymerase. It's a larger, more complicated entity that does things in a more complicated fashion. The stages of transcription are the same, the 4 stages, but each stage has more complexity in eukaryotes compared to prokaryotes. So once again, here's a summary of transcription comparison. So promoter consensus sequences in eukaryotes will be more diverse than in E. coli. Eukaryotes will have 3 different RNA polymerases compared to the 1 in prokaryotes. And the molecular machinery that initiates and continues the elongation of transcription is going to be much more complex in eukaryotes than we've seen in bacteria in this video. Thank you very much.